Hi, everyone. Welcome to the Biological Network Synopsis session. I'm going to co-host this with Woong-Yang Park, who is with me here. Today we'll hear about updates from some of the networks. But I also encourage all the coordinators to please send your summaries of yesterday's breakout sessions. You have received an email about this, and those summaries will be posted on the event platform and will be available for 1 year. Please do that, and so we'll get updates from all the biological networks. First out today is the oral and craniofocal biological networks, led by Kevin Byrd from University of North Carolina, Chapel Hill. Kevin, please go ahead.
Thank you so much. So I'm here today to update the human cell atlas on the progress we've made on the oral and craniofacial bionetwork. Our network is quite new. We established ourselves in September of 2020. However, unlike many of the other networks who may be loosely working together before formalizing, we actually just started working together during the pandemic in March. And so I'll show some of those updates just during that time and where we hope to go. I'll also put a plug in. I'll also put a plug in for our oral and craniofacial bionetwork symposium. The networks are shared between oral and skin. This will be on July 8th coming up quite soon. Our speaker will be Paul Sharp. We'll be speaking about both mouse and human single cell data. That'll actually be a really nice comparison between the two sites, as well as a skin network with Magnus Lynch. We find these sites to be really well collaborating already. Our core coordinator, Inez Siqueira and Masanifa, submitted a CZI proposal. And so hopefully we can see how these two networks are progressing in parallel. So as I mentioned, we established ourselves in September of 2020. I, in collaboration with Sarah Teichman and Blake Warner, started looking at the susceptibility of the oral cavity to infection by SARS-CoV-2 in the COVID-19 pandemic. At that time, we just had a few data sets we were aware of internationally. And so we brought our data sets together from both the gingiva and the gland. We integrated those. We'll show this data a bit later. And then we brought Inez, who's a longtime colleague of mine, to co-coordinate and found the network. Since that time, we've expanded from being about 4 investigators to being a little bit over 25. I say a little bit because I think at the meeting we've recruited a few additionals. The nice thing about our network is that while it might be a bit European and North American focused early, we are intentionally expanding already and reaching out to investigators in Africa, in Australia, yesterday in Brazil, as well as even just a conversation last week that I had with someone from Japan. Right now, we're fairly sex and gender balanced. About 5 out of our 25 are senior investigators, meaning that we're really heavily led on being early career. Right now, as I mentioned, that we have about 6 countries represented, though I think maybe after the meeting a 7th. And we're growing at a rate of about 3 investigators into our network per month. I think that will slow down, but right now we're fairly heavily focused on clinicians, as well as looking at basic scientists or translational scientists. We're really lacking, I think, currently, and one of our key features that we're going to need to overcome is going to be on the data science side. We really need to partner with other networks as well as other data scientists to achieve, I think, both integration as well as maybe more meta-analyses considering these sites. We successfully put on our own symposium to bring all these investigators together. It was an introduction to our network on June 10th. We had about 100 total participants. It was all virtual. About 15 presenters, many of which represent the group that you can see before you. When Inez and I were writing the idea of what this network was going to be, of course, we had the idea of mapping oral and craniofacial cells, which that will be defined here shortly. But also, we know that oral and systemic health are oftentimes separated. This is just a feature of very long traditions. And our goal is to hopefully, while we bring these data sets together in the idea of oral heterogeneity and craniofacial heterogeneity, to bring these meta-analyses to the rest of the body now. And so to do so while everybody's participating and bringing their networks together, which some of you might have received an email from me with really that idea in mind. Just as an update, what we have so far, up until the later part of 2020, there was no published oral data set, except for one publication on head and neck cancer back in 2017. Right now, in terms of the healthy reference atlas is what I'm showing here. There's a ton of oral heterogeneity that I think is underappreciated. And a lot of these sites have unique cells that we're already discovering that don't seem to have any other surrogate throughout the body. And so some of this uniqueness is something that we think is not, I think Aviv put it well, they're both rare, but they also might be rare based on their site within this niche. Among the oral sites in the major glands, we've got about 12,000 cells. If it's red, it's currently unpublished. Within the minor gland, we've got about 50,000 cells. Within the tongue, we've got about 10,000 cells, and then some published data on gingiva, even using different dissociation methods, I think in aggregate show about 50 cell types in this unique niche. And nearby that in the tooth is the pulp, which has about 40,000 cells. We've only integrated 2 of these sites so far. This was a collaboration I mentioned with Sarah Teichmann and her data scientist, Nick Huang. And we think we can do this now as we start to get more sites sequenced, but also pulling these data sites or these sites together for more data. We also have an interest in really thinking about what does it mean for the human cell atlas to consider how we study oral and craniofacial biology. The mouse has been a phenomenal model organism for us. We're also pushing towards organoids. And so thinking about the human as our central launching point, and then thinking about how we might test those things mechanistically, we started to put together integrated atlases for what is published for mouse data. This includes 2 glands and about the tongue and the mucosa. Other sites have been recently published, but currently we're still working on figuring out exactly when those are going to be published and bringing those investigators in. Subsequently, we've also, as I mentioned, integrated 2 sites. This integration has been successful for us in showing about 33 unique cell types. We think there are more, especially as you pull the niches apart. And if you look at each niche, we really understand that there's actually cells that kind of get muddled in the integration, particularly on the epithelial heterogeneity side and the barrier. And so we're thinking about looking at this both in aggregated, but also in individual atlases. As I can say for our network moving forward, being that we're in a nascent group, we need more cells. We talked about this yesterday and on Monday. We need more sites, and philosophically, we need to define what sites are appropriate for that first version of the atlas. And then we also need more integration. And by integration, like I mentioned, it's not just integrating among the oral cavity, but among the rest of the body. And so our first idea of this was a CZI proposal that we put in. This is an outline of what there were actually investigators on the lung Bionetwork side, and then I sat as an interface between our oral network, and we submitted a meta network grant. And the goal of this was really to consider the oral cavity and the nasal cavity as part of a bifurcated inhalation tube, which all connect down to the lung. And thinking about nasal biopsies coming from the oral cavity, and then how that might push fluids and tissues and cells and enzymes all deeper into the lung. We also are very interested in the fluid side. We think that these spaces communicate, and COVID-19 really revealed this to us, how these cells might be able to communicate, fluids, enzymes, all pushing kind of in this harmonious way of communication. And so as we think about moving forward, we would love to do so in collaboration with other groups, considering that most of the heterogeneity of the body is kind of packed within the oral cavity. And so we look forward to collaborating with all of you moving forward. Thank you for your time, and I'm happy to take any questions that we might have.
Thank you, Kevin. So we have time for a few questions. Maybe I can go first. In starting to make an atlas of such an heterogeneous place, and I imagine also a place where there's a lot of challenge in obtaining high quality cells. What are the greatest challenges specifically for the oral cavity, you think, compared to other tissues?
Yeah, it seemed to be that getting high quality samples might have been a challenge for some of our international partners. I can say that due to some early work that was already being done at North Carolina, we were able to partner with the lung group. We already had IRBs to obtain from ENT doctors, who one of those is one of our partners, was able to get us most sites. And so, for example, even something challenging like the muscles of mastication, which help us chew, developmentally, they're very unique from the tongue, which come from different developmental organs. Just based on different procedures and protocols already in place, we're able to team up and get those cells and get those to investigators. Some of those early partnerships have happened at Duke University, where there's a really fantastic muscle network. And so while we didn't speak about muscle today, I think it's actually a rich place for us to consider, again, that heterogeneity. And we think we have access to that But I think it's really getting the funding early. We never had a seed network. And so a lot of this is being done kind of almost thrifty. And we're doing so in a way that I think as we move forward with kind of organized proposals, you know, thinking about the muscle, thinking about how the immune sites are quite unique, and connecting that and even perhaps back to interactomics. You know, the oral microbiome is quite heterogeneous based on niche. The mucosa is different based on niche. And so we're interested in how that those unique cell types might instruct their own immune cues. And I think right now we've got the access, but I think the questions and the funding probably are our biggest challenge moving forward.
If I may follow up, then how about development? Are you going to address development? I think especially facial development is kind of unique and very interesting.
Yeah, you bring up a great point. We brought in Ophir Klein in their lab at UCSF. I mean, part of him and a lot of their partners there, they have one of the best in the United States of craniofacial centers. And they see a lot of really unique and rare diseases. And so while we're interested in oftentimes selling in grants that, you know, this is a common disease, it has a large global health equity need, you know, periodontal disease is quite common, cavities and caries are also quite common. I think there's a real need for us to be considering, I think early and often, the idea of rare oral craniofacial manifestations, perhaps on the developmental side early. Many of those individuals, if they do survive, they still have challenges moving forward. And Inez has thought about this with Muz and Aoife, because they were thinking about oral manifestations of epidermolysis bullosa. We have other ideas on pediatric IBD that have tons of oral manifestations. Again, it's a connected tube. And so I think making sure that we're considering the oral cavity as part of the whole body, developmentally, but also as pediatric diseases transition to adult chronic diseases is a lot of our focus. And so we'll have to intentionally reach out to partners to make sure we get those questions have been asked and hopefully answered.
All right, thank you so much. That's great. So I'll give it over to Woong-Yang to introduce the next speaker.
Okay. The next speaker is from the Baylor College of Medicine, Chen. You're gonna cover about the eye, right? So please start.
Anyone? Okay, cool. Good morning, good afternoon, good night, everyone. And thank you for the opportunity to give a quick update of the eye network. So I will first update you guys on the status of the current Cell Atlas project, version 1 from the eye network. And then I will discuss, recap the main challenges and future plan we identified through yesterday's discussion by network. So the human eye and our eye is a camera eye, and then there's really two parts. One part is functionally. The first part is where you capture light and then sense the light and then convert that light signal to electric signal and send that to the brain. So that's the retina, neural retina, and then the rest of the part is basically optical compartment. And then because of that reason, the network are separate into two parts. One is focused on the retina, this nice sensitive part, and then the other part, the rest of the part. So there are currently two CGI founded seed network. So the first one is led by myself, focused on neural retina. But the other network is led by Dr. Josh Chen at Harvard and focused on the rest of the eye tissue. So I will talk about the retina first, which is, as I said, at the back of the eye where it sends the light. It's a multi-layer neuronal tissues, and there are basically 6 major cell types, and neuronal cell types, and then a bunch of non-neuronal types. Although the number of major cell types is relatively limited, like about 10, but if you look at the subgroups, it is very complex. So based on the morphology and the electrophysiology started before, and it was estimated for human eye, there's probably like 50 or 60 subtypes at least. And one of the major challenges we are facing when we try to profile or build the version 1 of the retina is that not only the number of cell types is high, but also some of them are really rare. For example, the ganglion cells, there's about 15 cell types at least, but together they only count for 1% of the neuron in the retina. So our main challenge is how do we identify those exceedingly rare cell types in the retina. So we initially did a quick pilot phase to look at the cell composition of the retina, and we start with 6 donors, and then together we generate about 280,000 cells. And this is the T's name UMAP. Look at them, and then you can see we can identify all the major cell types. And to really build this map, because of this very exceedingly rare ones, we actually employ some enrichment method, try to normalize the cell type as much as possible. And therefore, we can, for example, you can see the RGC, we bring it up in this data set to like totally about 3%, from less than 1%. So from this pilot phase, we identify 13 bipolar cell types, 34 amicrine cell types, and the 9 RGC cell types. So based on the previous knowledge, we know the current sensitivity for this particular version is around 0.01%. So if cell more than 0.01% of the population, we actually detect as a cluster in the map. So and during the process, there's several data set published by the community, and we start reaching out to them. And then the idea is that now we know although we have all the major cell types, but we're probably missing some subtypes. So so we crank up our production by getting more additional donors and then provide more data. So in our group, we generate about 1.7 million cells at this point. And then together with the published ones, about like 250,000 cells, we have about 2.1 million cell total from human retina. And work with the SAS group, we are starting to try to integrate those data set together. And then this will be the backbone for our version 1 human retina. So you can see we already find all we'll be able to integrate all the cell types together. And then this is the cell numbers, and then the numbers that we were high, except in RGC is still quite rare, it's 40,000. We are trying to right now to specifically focus on these cell types and then try to increase the number a little bit more. And that will be the final data set. So this data set, we are working currently with Stanford group, and then HCA try to deposit it before publication. So move on to Josh's group, and they also did a fantastic work on pushing all the other parts and profiled. And then those are the anterior segment of the eye. You have the light coming from here, the retina is down here, and then you have the cornea, and then the drainage system, the lens. So you can see there's all those parts have been profiled. Some of them already published, and some are in preparation. The data has been deposited. So with this in hand, and then Josh actually pushed into a comparative study where with the retina data in hand, and then the profile three additional species. And then what's interesting is when you compare the cross species, you can see the most conserved cell type are photoreceptor cells, the cell that capture the light. And once with you moving down to the layer, and then for example RGC is where the cell that connects to the brain is much less conserved. So and then that underscore importance to directly profile human, and in addition to the study of other model system. And similarly, the anterior segment actually quite conserved across species. That the same the other model system will be really useful to study the other parts of the eye. So just much as I said, that this data has been deposited. So we're in the process of being depositing. So those are the list of some of the data set. So the major challenge we identified from yesterday's discussion is, because there are many active groups are producing data, in addition to the CGI network, and so how to compile and integration this data set generated from multiple groups with different technologies is still quite challenging, particularly when the number of cells grow big. Like in the case of Retina, the 2 million cells, many of the integration software does not work. So, and the second thing is how to proper, this is probably even harder, how to properly annotate a large number of cell types in the tissue. Many of the cell type has not been started before, and then to even have a consensus naming, and then sort of correlate with previous naming is quite challenging. And how do you distinguish cell type versus cell states? That's another question. And there are cells are moving at different physiological situation. and lastly, the easy access process, the reference data set for researchers who are not bioinformatician is actually one thing high on the list. So the major needs we identified is we need to have a data set to cover variation in human population, such as age, gender, and ethnic groups. That's the network trying to do, and then that's what the current data set is sort of lacking. And another thing is we have a lot of data for the retina, and then however the other region of the eye is much less profiled, so we need to increase the effort over there. And lastly, we also start generating some single cell spatial transcriptional data and also epigenetic data that should go along and integrate with transcription data. So that's area which is very little data at this point. So the future plan as the network, we would like to grow the network. So we are reaching out to global community and then grow it. And then we are starting to organize the regular meeting and activities. And we would like to take advantage of the HCA infrastructure taking place that give monthly seminar, it's also for and interact with other network. So that's in plan. So we since the diversity of the tissue, the organ, so we decide to form a working group with focused tasks. For example, one of them will be organize a community annotation effort for the Eye Atlas version 1. Even now we have the clusters, how to name them is still something ontology we need to figure out together. And also we would like to extend our network to include not only the adult data, but also development data, which is there's some out there already published and what people have in their hand. And also include them as a part of the reference data. For that one, also most of the development are studied done on the retina, but the rest of the body, the rest of the eye is pretty much no data. So that's one thing area we need to pay attention to. And then lastly is broadly distributed reference data in both raw and processed format. So we are depositing to HCA site and DCP and then cell by gene, but also there's an NEI and National Eye Institute have the effort to put database together. So we're going to deposit in their places so make it more visible to the community. Thank you very much for the extension, and then please join us if you're interested by emailing humancellatlas.org.
Okay, thank you, Dr. Sten. Maybe we have a question from the audience, the Gama Akabani, and then he asked about, do you have comparative studies with the eye of the mouse? If yes, I add so, one more species, we ophthalmologists actually they use the rabbit model.
Yeah, so the current comparison is done between human, rhesus macaque, mouse, and then also for retina, there's a chicken and other species.
Okay. Yeah, and for my side, actually, you use that nuclei for anterior part of the eye, and then retina will be just single cell, right?
So for the retina actually is primarily single nuclear RNA-seq. There's some one of the data set is actually single cell RNA-seq. For the anterior segment, currently it's a single cell RNA-seq. And then when Josh's group tried to expand to additional donors, they are switching to a single nuclear RNA-seq just because it's really hard, much harder to get a fresh tissue dissect and process.
Different structures and then Then maybe if I add one more thing, any chance to looking at the microbiome or other virus sequences together in the cell data, because it's gonna be there's so many viral infections or some it's not commensal, but actually maybe we can look at that together, right?
Yeah, for at least for the surface of the eye and tissue, definitely there's a microbiome play an important role. For some disease, for example, dry eye disease. So I know this group looking into that. For our group, we haven't not started looking at, but that's a really good idea. So we definitely should start looking into that.
Okay. Any other questions from? No. Okay. Then we can move to next speaker, Sten. Yeah.
All right. Thanks. So I'm happy to introduce the next speaker, Susanne Mandrup from University of Southern Denmark, who is going to present the Adipose Biological Network.
Do you see my screen?
Yes.
Yes, looks good.
Yeah. Excellent. So thank you very much, Sten. And I'm, as you said, going to present our status report for the Adipose network on behalf of Evan Rosen and myself. And we are at a really early stage in this network. We only really started to discuss earlier this year. And so far, we've not really done any collaborative work. So it's what I'm presenting is really mostly a collection of the challenges that we have in the tissue and how we are planning to solve this and how we are planning to work together. So in the adipose tissue, we have a challenge that there is a lot of fat. So the adipose tissue is a connective tissue. It's widespread in the body. It's associated with many different organs. So adipocytes differ between these different depots. And there's also a really ill-defined structure of the adipose depot. So the most adipose depot consist of these parenchymal cells, the adipocytes, that take up perhaps 20 to 30% of the cell number, but they take up most of the space in the adipose tissue. And these cells are really not compatible with conventional single cell analysis because these cells are large and too large and they're lipid filled, and all this lipid interfere with enzymatic processes. So this is our challenge. And if you do cell dissociation, you can see that the adipocytes will float, whereas you can precipitate the stromal vascular fraction of the adipose tissue. So many researchers have looked at this stromal vascular fraction, but it's only recently that we are now starting to analyze also the adipocytes. And the trick to do that is to do nuclei isolation, whereby you can get a relatively unbiased collection of nuclei from the entire adipose tissue. So the challenge is then when you do nuclei isolation is, of course, that there is an increased amount of ambient RNA. So you need to maintain a good nuclei structure and a good integrity of the nuclei, such as little ambient RNA as possible. And our tissue also has particular challenges with respect to spatial transcriptomics, again, because the lipids and the big lipid droplets in the adipocytes take up most of the space. So you can only really detect transcripts at the rim of the adipocytes and in the nuclei. So those are challenges we're trying to solve, and we're actively discussing in the network. So previously, there's been a number of publications on specific cell populations, mostly the progenitor cell populations, the fibroadipose progenitors, and the immune cells in the adipose tissue. There's a lot of different immune cells in the adipose tissue. But it's only this year that we have the first complete map of cells in the adipose tissue. So we published earlier from mouse epididymal adipose tissue a map of all the different cell types and the plasticity of these in response to obesity. That was in mice, and so far there's been no published account of adipocyte and adipose tissue from humans, but Evan Rosen has something in the pipeline for publication, and I know several others have also stuff that are working on. So we can see it is possible indeed to get an overview of all the different cell types in the adipose tissue at once, instead of having to split up the tissue in multiple fractions. So as I said, we only really started to discuss this year, and we have had active discussion with Christian Wolfram and Timothy Sullivan and Patrick Seal, who offered to help in this network. But we're really interested in hearing from others who want to join this network. So please write to us at this email address here, adiposehumancellatlas.org. And based on our discussions yesterday and at previous meetings, we plan to take a first step by recruiting a group of postdocs or staff scientists from our individual labs with some computational skills, so that we can do initial comparison and integration of data across our labs, across different depots, different methods, different physiological conditions, obesity, diabetes or not, and do this for both mice and humans. So most of the data that has been generated so far has been generated from mice, and we believe that we can really learn a lot from data integration from mice that can be later on applied for studying human adipose tissue. So one of the first things we want to do is to identify a common nomenclature. It seems like everybody who has done single cell sequencing in the adipose tissue have invented their own nomenclature, especially for the fibroblastic and progenitor cells. So we need to come together and find a common nomenclature. We also want to establish a homepage to increase network identity and for dissemination of our data. And we haven't planned workshops yet, but we really would like to do that in the fall. And then we think it's really important to look for funding for generating more comprehensive reference maps in the spirit of the Human Cell Atlas. We cannot, with the manpower that we have in our own labs, do this. So we need to look for additional funding sources together. So this was all I had. And as I said, we're really, really interested in hearing from others who wants to join in and provide some help in these first initial data comparisons integration. Thank you very much for your attention.
Thank you, Susanne. Just what about development? So have you started thinking about that? And are there particular challenges in terms of development of these tissues that are sort of diffuse and less of a distinct organ?
Yeah, it's not something that neither Evan or I are particularly interested in, but there are other members of the network who are interested in that. For example,
Okay, next speaker from Imperial College of London, Dr. Michela Noseda will cover heart Bionetwork. Thank you. Yeah, please, Noseda.
Thank you, Woong-Yang. Here I am. Let's try with our slides. I'm presenting on behalf of our network. And here we are at the heart of the HCA. The heart is a really amazing organs, which never stops working, otherwise we die. It beats about 100 times each day, and it needs to continuously pump about 7 liters of blood throughout the body. It's composed of 4 chambers, which withstand very different pressures, and you can imagine they have to pump the blood in different districts of our body, or receive the blood from different districts. The heart has an estimated 2 to 3 billion cells. I think we've done a good job in starting profiling a good amount of these cells. The cardiac cellular landscape is complex, but we're seeing that any organ is really composed of some unique cells with specific functions. In the heart, the cardiomyocytes and the conduction system are probably the most specialized cells. And then we got the support cells, the immune cells, the vasculature that are common across all tissues, but that we know have specific functions in each of the organs. Now, talking about cardiomyocytes, this is one of the challenges that we face in profiling the heart. The cardiomyocytes are very large cells. They're actually almost like a rectangular shape. They're about 100 microns long. So they're different to profile as a cell because they won't go through your typical microfluidic platforms. They can be faxed. So there are technical challenges in this that we have started to overcome by combining single cell profiling with single nuclei profiling. And of course, the network is starting to embrace spatial transcriptomics much more actively. So the network has, I would say, has really boomed. In October 2019, in the HCA meeting, I think I was one of the people reporting in that case. We had 2 projects registered. Now I think it's 189 participants. I can't read the top of my slide, but if I remember correctly, it's 189. So a lot of people. What is really encouraging, we got contributors from all 5 continents. Some areas are more, have contributed so far more, like North America, Europe, but we also have data deposited from Africa, South America, India, China, so Russia, very, very, very encouraging. We're never enough, the more the merrier, so please join our network, contact us at the email indicated at the bottom of this slide if you like to contribute your data and work together. The network has been quite productive, especially in the last year, despite the pandemic. A number of papers came out. The network has contributed also to COVID, work on COVID. So the various papers that came out, the TACE paper, what we call in the network, it was the first paper analyzing single nuclei from human heart. Our work done in collaboration with Sarah Teichmann, John and Cricket Seidman, and Norbert Hubner and many others managed to collect data from 14 hearts. So these are donor hearts, as you can imagine, very precious, and we're very thankful to the families of the donors that allow us to do this work. We profiled 7 males and 7 females and generated an atlas that is results from the combination of single cells and single nuclei. Only our paper has collected about 500,000 single nuclei and single cells. Within the network, based on the HCA data, there are over 800,000 single cell and nuclei data available. As I mentioned before, we have, as a network, we have contributed to mapping of the infamous ACE2 receptor in the heart. This is predominantly expressed in the pericytes. We do a lot of work in validating by RNA scope our findings. This is just an example. Now, what's on the agenda? What's next? Well, from discussion in the booth and in the breakout session, I think there are 4 major action points or goals that we want to achieve. One is increasing the ethnic diversity of our samples. If we think to just referring to our work with 14 samples, those are all white Europeans. There was not a single there was really no diversity in the ancestry of these patients. The age is another point that we need to expand on. Most of the donor hearts that you end up, at least for the healthy samples, the donor hearts are often from hearts that cannot be used for transplantation. One of the criteria for a heart to be rejected from transplantation is age. So you end up having a population that is a little bit skewed, and we don't have much below 50, 40 years in the adult. Anatomical regions, sorry, I didn't mention it before. The most regions so far that have been published is the 6 regions, 6 anatomical regions in the heart. But as I mentioned, the heart is a very complex organ with many different functionally specialized area, and we need to move on sequencing those. Another point of discussion has been the need for deeper sequencing, at least in a subset of the samples. Now, what are the challenges that the network as a whole has been encountering, still is happening? So we were successful. Many of us published data on the healthy. We're starting to profile disease hearts, but tissue procurement remains a major obstacle. At the moment, the way we collect data is via organ procurement agencies. So donor hearts, as I mentioned, that cannot be used for transplantation for a number of reasons, are donated for research. There has been a suggestion or I don't know, of trying and using more rapid autopsy programs, getting access from tissue programs. What we can then use, and we have been using, is cardiac explants. So these are the heart, the disease heart that gets removed from a patient at time of transplantation. We use the apical core at time of LVAD implantation. LVAD is the left ventricular assisted device, is a mechanical pump that is inserted at the apex of the heart in patients with failing heart. So when they insert this mechanical device, they remove some tissue that we have been using. What I labeled as leftover tissue is basically that's what the surgeon I collaborate with, he called it leftover tissue. If it's removing part of the atrial appendix to insert a catheter, that is basically tissue that would just be disposed. It gets passed to us to do some profiling. Biopsies in our field are very limited. They are not done routinely as happens in cancer, so there is some access, but very limited. The other challenge I mentioned before, we want our atlas to be more diverse. At the moment, there are a number of roadblocks to achieve this. And it's very important that we establish collaborative projects with underrepresented, sorry, I misspelled that, underrepresented countries. And the limitations here are beyond the scientific interactions. What do I mean? I mean that you can find collaborators in underrepresented countries, but what is not easy is to actually implement the collaboration. There are countries in which laws, government, religious, cultural, political influences really block the export of samples and the sharing of resources. Third point that we challenge is the one aspect raised by Nathan Tucker and others agree is the issue regarding open access of data. So there are certain populations that might be willing to donate their tissue, but they are not very comfortable with the open access of the data. So it's something to discuss with the ethics group, the equity working group, and any other member of the HCA that might help us out with this. So what are the action points to try and tackle some of these challenges are, on one point is community engagement. So what we refer here is really a broad and intense effort. We think it's necessary. Engage with the general public, especially engaging populations that are underrepresented. If we want to achieve ancestral and ethnic diversity, we need to target specific populations. If I think about London, London is 40% BAME, but still you never get a non-European background heart donated. So there are issues, cultural issues, religious issues, and it's not something that we can change overnight, but we need to really engage more. To increase the number of organs available, we need also to actively engage with donor procurement agencies and working very closely with clinicians who are our point of contact with patients. And this includes pathologists. We're very lucky in our network. Mark Alushka is part of the network. He's the president of the Society of Cardiovascular Pathology and is very keen on create a good connection with the HCA Heart Network and pathologists, especially pathologists under training, and make sure that we connect the dots there. The second point here on the slide is about, we said, collaboration and sharing resources. This is more complicated with some, in some circumstances, with some countries. And one of the one points that we raised, it's important that funding agencies understand that we need much more time to establish certain collaborations than we might think. If we think about the CZI ancestry call, it was great, but when we engage with some countries, we realize time it will be exceeding the length of the grants by the time we can actually use collaborative some tissues, and had long discussions with Partha Mojambe from the equity working group, and really we might need to really dedicate resources and personnel to make sure that we can go through government processes to allow this more open use of resources from, as I said, certain countries.
Michela, actually, we have 2 minutes, right?
Yes, we're almost done. We said if we can work together with the ethics working group and equity working group to revise the open access data regulations, and a general comment to the Heart Network and possibly to any network, we all need to make a big effort to ensure we collect metadata and we share metadata in a very organized way to allow deeper analysis in the healthy and disease samples. Thank you very much. A special thanks to people who helped with the slides, Mona, Enrique, Alec, and Susanne. Thank you so much.
Okay, thank you, Michela. Actually, there's many questions for your presentation. Actually, if I summarize, they interest about some special tissue for the the conduction system is the electric conduction is most important. So, any chance to looking at that electrophysiological the phenotype in the cell level?
Yes, absolutely. We are starting to collect these anatomical regions. So yes, it's work undergoing, essentially.
Okay. And the other question from Bruce Arnold, he's interested about pericardium. So maybe it's some related to some diseases or some protection of heart. So
yeah.
Do you include that the pericardium tissue? Also in that, I'm just about the valve.
Yes.
Another, yeah. Important.
Yeah, absolutely. As I mentioned, there are so many anatomical regions within the heart, and that's exactly what we're doing now, expanding and doing much more targeted and patient dissecting of the heart. This is only possible though when you are allowed to work with the whole heart. So you will get the whole heart, and then you can actually sample the regions of interest. Some instances, ethical, the protocols allow only to take some. But yes, we're working on all of those. The pericardium is a good point. I mean, already in the data published, we do see epicardial cells. But the pericardial sac is a good point that was raised, and we should start sampling on that as well.
Okay, thank you, Michela. I'm going to move to next final speaker.
Thank you.
All right, thanks. Our final speaker for this session is Villani from Mass General Hospital, who is coordinating the immune system network.
Thank you for an introduction. Greetings, everyone. I'm delighted to share with you some summary of our breakout sessions for the Immune Network, and I'm speaking also in the name of Nir Heiko and Tom Tiggin, who are co-coordinators of this network. We now have 75 participants in our network, and having a lot of us have independent efforts in generating large amount of immune cell data. And so what we discussed during our breakout session is a plan over the coming year through a series of events and meetings to reach a detailed roadmap for our immune cell atlas, which is what I'm going to review today. So you're all aware of these series of events if you are interested in participating. And I'm going to right away mention it, and we'll mention it again later. If you're interested by what I'm talking about, please join our network by emailing at immune@humancellatlas.org at the bottom of this slide. And so quick highlights of what we're going to be discussing. So we discussed a new immune bionetwork seminar series, a series of meetings around developing framework for annotations. I'm going to go in details for all of these meetings over the next few slides. A Jamboree event to test and optimize our annotation frameworks, and finally a detailed roadmap to be posted by the end of the year. And so in terms of community event, these monthly seminar series will start in the coming month, hopefully, and when everyone is invited to join, we'll try to host 2 labs per month, and we'll be alternating events morning and evening to accommodate different time zones. We also realized that we don't have a very good inventory survey of all of the ongoing immune bionetwork projects, which is a bit of a problem to establish our atlas because there's so many efforts across the immune system, which of course encompasses all tissues and organ system. And so for those of you that have signed up to our network, you'll be receiving a survey in the coming weeks where we're going to try to gather information about the different projects, data set, metadata being gathered, and that information will really help in prioritizing the first draft of our atlas. And our first annual meeting, we're hoping to host in early 2022. It may be a mixed medium of in-person and meeting with virtual component. And the primary goal will be centered around subtype annotation. So let's dive in a bit more in detail. So we have a detailed plan for our meetings, which is what was discussed during the breakout session. And so for the first 4 4 meetings. So this is separate from the seminar series. This is on a volunteer base. We're gonna invite 6 to 10 groups to participate. And the goal here is to present your approach on immune cell annotation. Every meeting will be about an hour, and we're hoping to have 3 groups present. The primary goal is not to present your specific biology or findings you have made. The goal is to present your cell annotation strategy through the lens of your projects. This includes upstream data processing, which features need to be prioritized that are most relevant to immune cell type annotation, because across all networks, we are all facing challenges with annotation, but some are very specific to the immune system. For example, the use of TCR data to map specific T-cell subgroups. We talked about mate cells, the use of antibody-derived tags to help prioritize markers for splitting lineage and mapping cell states, and so forth. The outcome will include, after each session, a summary of the different strategies will be posted in a Google Doc and open for comments and questions. Following up to these first 4 meetings, we'll follow up with another 2 meetings. And of course, these meetings, time allocated may vary depending on feedback. Will involve summarizing and discussing the main framework for data annotation and cell type state discoveries. This is open to, of course, all Bionetwork members. We also want to involve other Bionetworks members that are interested by immune cells, and we will want active participation and feedback from computational experts. And we're thinking of 2 meetings of an hour and a half. The primary goal is to present a summary of all the clustering and annotation frameworks presented. We want to discuss what could be reasonable parameters to standardize our analysis and annotation frameworks. We are not looking for a one-size-fits-all solution. We are considering using several annotation strategies in parallel. Could be 8, 10 annotation strategies in parallel. Some could be specialized to specific modalities of measurements, and we can implement a ranking matrix consensus frameworks with cross-cutting terms. And then manual curation could focus on disagreement between annotation frameworks. We could consider a semi-automated approach that can involve experts helping with manual curation. And the group all agreed that until we have a solid first manually curated immune cell atlas draft where experts have been involved, we cannot simply rely on just machine learning frameworks. We don't yet have a very good curated draft. We also discussed what type of optimal platforms could be used to empower crowdsourcing annotation. So we talked a little bit about Peter Karchenko's platform as an example. And so the outcome would be a set of general recommendations for annotation, again, summarized in a Google Doc documents that would be open to all for comments and over the course of a 2-3 week period. We want to follow these 2 meetings with a jamboree that will test these annotation recommendations. These will be open to all immune Bionetwork participants. We're seeking for volunteers and have groups made of computational biologists and immunologists to participate to the event. As I mentioned earlier, it would be a 2-day meeting, but we could have a virtual portion to make sure that access to the meeting is as democratic as possible. And so to test and optimize our annotation framework, we're proposing making a data set of several combined COVID-19 data sets. There's been so much PBMC's data sets published as part of this unfortunate COVID-19 pandemic. There's an opportunity to leverage these data in this case. And we're looking to combine them because annotating sizable immune data set is quite challenging compared to small data set, because the bigger they are, the more cell states you can uncover. And so upfront, this is something that was brought up yesterday. We'll want to involve teams of computational experts that will help with data integration and initial clustering of the immune cell lineage, because ultimately we're more interested by subclassification of cells. The exercise itself, team will be given a day and a half to annotate different cell types, and depending on the number of teams involved, that can involve the entire data set or a specific cell lineage. And each team will have to present their annotation by day 2. And by the end of the meeting, we would compare the annotation solutions between groups and discuss source of discrepancy. And hopefully the results will help refine our proposed annotation framework. Some yesterday suggested considering comparing our resulting manual curation to automated annotation to show how our approach can improve automated annotation. And so the outcome will be a summary of the Jamboree, which eventually will be reported in a white paper, and we'll also make the results available on the data portal to empower the community to give feedback on annotation as well. And upon summarizing and updating our annotation framework by meeting 7, we want, through meeting 8, present this annotation framework to a broader immunology community, across experts across many different lineages to get their feedback. And so the scope would involve meetings with different cell lineage experts, and the exact time allocated is to be adjusted. And it is important to get feedback on our annotation framework recommendations from these immunology experts, because we need the buy-in from the broader immunology community if we want them to ultimately use our data as a resource. We need to figure out how to best integrate legacy knowledge of the field of immunology in our annotation. We're not here to reinvent the wheel. We need to make sure that whatever we put together is well in line. And we will, in that spirit, we will also make our Jamboree data and results available on a platform, as I mentioned in previous slides, to enable crowdsourcing feedback on our annotation by the broader community, and that may help us fine-tune our recommendation. And again, this is always in the spirit of trying to get to a more consensus approach for these annotation. And so we hope that this is going to lead us to a final set of meetings to develop our roadmap. This is going to be with the scope of creating this white paper. And so this is going to be open to all Immune Bionetwork and representative of all Bionetwork that are interested in taking part of drafting the roadmap. We're envisioning 3 to 4 meetings together with offline work on drafting it. And we'll be dividing the sections across participants involved. And so that would include some of the guidelines and plans for integrating different data sets and efforts. We'll also describe and discuss recommendations on different critical experimental and analytical topics, as well as annotation recommendations, which we discussed yesterday. So some of the topics included sampling and measurement strategies. So these are the questions that are, over the last few years, our network has been discussing. So how do you capture all rare cell states within a frequency of 1%? How many individuals do you profile for a first draft across gender, age, range, ethnicity, and geographical locations? We know that immune cells are defined by their functional response to immune challenges. So how should our measurement focus on healthy versus challenged states? Challenged states here being disease. What is the minimal set of modalities of measurements that are needed to define new cell states? Do we really need all these modalities for first draft? And how do you know that a predicted cell state is actually meaningful biologically in immune cells? Because they're so plastic, this question is particularly important to tackle. And from the analysis standpoint, as you know, a lot of the meetings that were just showcased minutes ago have to do with harmonization and annotation. So we're going to describe our results here. That's important to figure out if we find the same immune cell states across different tissues and studies. We need to figure out a way to harmonize our nomenclature as we name new cell states and integrate historical nomenclatures. We're going to describe how we can best empower every tissue network to contribute to our immune bionetwork, and how we can develop a user-friendly online interface to empower immunologists around the world to explore and query our data and contribute to annotation. So we're looking forward to working with Peter Konchenko and others that are working on such platform and describing what is our first immune draft. And in parallel, this doesn't need to be meeting 12, this will happen in parallel. We'll be working on the first draft, and so all key stakeholders interested in participating are welcome. We're going to identify the groups of immune cell data set that could benefit from being integrated and merged, and this is going to be based on our survey. And then we'll proceed on harmonizing this data annotation and subdividing the merged data set based on cell lineage so that we can divide and conquer. And one of these efforts could be an extension of the January and COVID-19 PBMC's data set. And the outcome will involve uploading the draft on the web portal to empower community-based feedback and annotation, and of course also to empower the community to query the data, and a series of manuscripts detailing how we have merged the data set and annotated it. So with that, I'm out of time. I'm just going to put all of the other topics that were discussed, saying that some organs including lymph nodes that will involve analyzing the cells without beyond immune cells. We need to bridge with other biological network. We discussed second-tier analysis involving transcriptional programs and analyzing mouse in parallel so that we can have a mouse-human mapping, and that the initial draft will not be perfect, and that it will continue evolving as more data modalities are integrated to include disease data, and finally to work on harmonizing the metadata. So with that, everyone is welcome to join our immune network. We need everybody to make this first draft. Please join by emailing us at this email address, and we'll be in touch with specific dates. Thank you.
Thank you so much, Chloe. Indeed, we're running out of time, but we have time for one question. This is from Kirsten Meyer in the audience. The question is, will your first annotation include infection and disease, as this is normal for the immune system to reveal all the cell states?
Absolutely. This was quite a high point of discussion yesterday. We strongly believe that in our network, to capture the full breadth of immune cell states, we actually need to look in the perturbation setting in disease. And so we are looking to integrate disease data set in parallel for the first draft.
And is there a systematic way of sampling disease?
Well, that was the whole question about metadata. And because not everybody is sampling disease the same way, and because we don't have a good idea of what are the different disease efforts that are ongoing, this is why we need that survey. And so once we have a very good understanding of the different projects involved, that will allow us to prioritize the first atlas. So please, all of you that are interested in the immune network, do fill out the survey when you receive it, and we look forward to working with all of you.
All right, thanks. And thanks to all the speakers, and thanks to all the coordinators of all the biological networks for the contributions. If you're interested in joining, get in touch. Email links are available on the Human Cell Atlas website. And I think that concludes our session.